Okay, so in the previous video, what we did was we saw how we took the starting material of N-acetylglucosamine from the cytoplasm of the bacterial cell, and we bound it to a uridine diphosphate molecule to get a uridine diphosphate N-acetylglucosamine molecule, so a UDP, and then we've got N-acetylglucosamine here. Okay, and from now on, rather than drawing the entire structure of this molecule, we'll draw this cartoon, basically, to denote it. Okay, right. So the next conversion that is going to happen is we are going to bind onto this UDP N-acetylglucosamine molecule. We're going to bind a phosphoenol pyruvate molecule to it. Okay, so let's go over the structure of phosphoenol pyruvate. It's basically a molecule that is produced in glycolysis of respiration. Here what we're going to do is we're going to add it on to our structure in order to convert our structure from being a UDP N-acetylglucosamine molecule to being a UDP N-acetylmuramic acid molecule. Okay, so phosphoenol pyruvate. Okay, right. This is often denoted PEP for phosphoenol pyruvate, and the structure of phosphoenol pyruvate is as follows. It's a very small structure, you'll be relieved to know. Here is the carboxyl group here, and then off this middle carbon here you have another carbon doubly bonded to it. That's where the enol of its name comes from. And then off this carbon you also have a hydroxyl group which is bound to a phosphate group here. Okay, so this is our phosphoenol pyruvate molecule. Right, what we are going to do now is we are going to bind this phosphoenol pyruvate group to our hydroxyl group of this UDP N-acetylglucosamine. So now let's have a look back at the original structure of N-acetylglucosamine. Off this third carbon of the glucosamine molecule, you had this hydroxyl group coming out of the page. Now, I may have gone to drawing this cartoon structure here, but that hydroxyl group is still there. And basically what we're going to do is we're going to perform a condensation reaction in which we add this phosphoenol pyruvate group onto this hydroxyl group here. Now, you might think, oh right, well maybe we'll add this hydroxyl group here and with this hydroxyl group and we'll perform an esterification reaction. That's not what happens. Instead what happens is you cleave off this phosphate group here. Okay, so you cleave off this phosphate group here. You cleave off the hydrogen from here. You put that hydrogen onto this free oxygen here to get a phosphate group back again. And then you bind this oxygen to this carbon here. Okay, so let's draw that structure that we're going to get on the next page, basically. Okay, so, right, so let's just redraw our UDP N-acetylglucosamine molecule first. So this is our UDP N-acetylglucosamine first. So we have our six-membered ring, which is our glucosamine, and we have our sixth carbon going off down here. We then have our amino group down here with its acetyl group coming off down here, and I don't know why I'm still drawing this one structure out when I'm abbreviating everything else's structure to just blobs. And here are these two phosphate groups, and here is the ribose sugar, which we'll denote as a pentagon here, and here is the uracil organic base there. Right, okay, so now what we have done is we've taken this hydroxyl group on the third carbon of the glucosamine ring, and we've bound it to this second carbon of our phosphoenol pyruvate molecule. So let's transfer this structure onto here. So here's the second carbon. It was double bonded to another carbon with two hydrogens coming off it here. And then up here it was bound to a carboxyl group. Okay, so here is what we have produced now. Now this is not yet UDP N-acetyl-muramic acid. Instead, what you have to now do is you have to reduce this double bond here, which means you break one of the bonds of the double bond and instead bind these two carbons to hydrogen molecules instead, hydrogen atoms. So we're going to break one of these bonds, and we're going to instead bind this carbon to a hydrogen atom, and we're also going to bind this carbon to a hydrogen atom. And that structure that you now have, that is UDP N-acetyl-muramic acid. So this is UDP N-acetyl, and now instead of being glucosamine anymore, it's now muramic acid. Okay, right. Let's have a look at this name. UDP N-acetyl-muramic acid. So, that means that if we took this UDP molecule off, what we'd be left with here... Okay, I'll highlight it. I'll get my coloured pens out. Okay, so this bit, with the UDP taken off, basically, that, which is the glucosamine molecule bound to this phosphoenol pyruvate molecule and then reduced, that is N-acetyl-muramic acid. So when people talk about N-acetyl-muramic acid, that's what they mean, muramic acid. Right, okay, and just like people denote N-acetylglucosamine as NAG, people often abbreviate N-acetylmuramic acid to to NAM, to NAM. Okay, and you will also see people referring to N-acetylmuramic acid as N-acetylmuramate. So, just like if we talk about glutamate, you can also use the expression glutamic acid. It's just like that. Instead of using muramic acid, you can use muramate. The strict chemical difference between muramate and muramic acid is that muramate would be the conjugate base of muramic acid. So the molecule I've drawn here is actually N-acetylmuramic acid because it has the hydrogen still bound to this hydroxyl group. So it's an acid which hasn't yet donated its proton. Now, if the acid was to actually donate its proton, which is what acids do, then the oxygen would gain a negative charge and it would no longer have its proton. That molecule would then be muramate, or N-acetylmuramate. This really is what's known as the conjugate base of N-acetylmuramic acid, because it's the acid molecule that has donated its proton. And why is it called a conjugate base? Well, if you think about it, what do bases do? They bind protons. This group now, which has this oxygen with this negative charge, this would want to receive a proton, so it's become basically a base. So all acid and base molecules have this sort of partnership where they can receive and donate protons, and when they've got the proton, they're an acid because they can give away the proton, and when they've given away the proton, they're a base because they want to receive the proton back again. So it's sort of like they can't make up their mind kind of thing. So that's what the strict difference between N-acetylmuramate and N-acetylmuramic acid is. But of course, if you have a tub containing some N-acetylmuramic acid, loads of the molecules in there will actually be N-acetylmuramic acid molecules, i.e. they will have their proton still bound, and loads of them will be N-acetylmuramate molecules, meaning that they've donated their proton. So any sort of solution of it that you have will always contain a mixture of both types of molecule, and therefore you can use the names interchangeably, basically. Okay, right, so far we have now made this UDP N-acetylmuramic acid, and now what we're going to do is we're going to add amino acids onto our N-acetylmuramic acid in order to create an N-acetylmuramic acid tripeptide, and then we'll create a pentapeptide. Okay, and we'll continue this story in the next video.